When studying cellular respiration, we want to look at a specific parameter, which is going to be the production of CO2 gas. In cellular respiration, organisms such as plants, animals, fungi, typically all cells have the ability to carry on some form of cellular respiration at a cellular level as well as an organismal level. And the reactants that go into cellular respiration are going to be glucose and oxygen. Once the reaction is completed, the products that are produced are two. You're going to produce water, which can be recycled in the cells or come out as water vapor. But the second very important product of cellular respiration is CO2 gas. And CO2 gas is something that is measurable in a closed environment. So in the cellular respiration lab, we're using these little, they're handheld computers basically. They're called LabQuest units. You may have used them at the high school level. We've had them for a couple of years. And you can see that it's measuring CO2 right now, and the numbers are all over the place. And the reason why the numbers are all over the place is because this is a ventilated room. So there's fans all over the place blowing the air around, and it's just kind of laying down, which it's supposed to be standing up. But I'm going to be calibrating the sensor outside where you have regular ambient CO2 levels. I'll insert it into the container and then start the lab collecting the CO2 data here inside the lab after I've calibrated it outside. Then I'll bring it in, set everything up, and we're good to go. So in this lab, we're using, these are respiration chambers, or what we all sometimes call them as metabolic chambers. And you can put in them any type of organism you want. Typically, we are going to be doing this lab with two different organisms. We're going to be using these are giant worms that are in here. They are larvae, beetle larvae, megaworms they're sometimes called, and for an animal. And then for a plant, I'm going to be using bean sprouts, what they call mung bean sprouts, because when beans first sprout, they don't have any leaves. So they cannot photosynthesize. When you think about it, when seeds sprout, they sprout under the ground. So they're carrying on respiration. Okay, they're not doing photosynthesis, they're carrying on respiration. So in a little while, I'm gonna go get some fresh bean sprouts, and we'll use that for the plant portion of the lab. But the way we set this up is we'll put our organisms in here. We know what the mass of each one of these Nalgene bottles is, so that once we put our organism in there, we can weigh it so we know the mass of the organism. And then I've already got my sensor connected to my LabQuest unit. And then what you do is you just insert the sensor into the unit, and then I'm going to program the mode, the rate, and the duration. We're going to run this experiment for 30 minutes. And you want to set up those parameters so that your LabQuest unit is going to automatically take a measurement, for example, like every 5 minutes. And then that will come up in a graphing data, so you'll have your time, like the CO2 at time 0, the CO2 at time 5, CO2 at time 10. And from that, we will create our data tables for this lab. So this is the kind of equipment we're going to be using. Now, we're also going to run this at 2 different temperatures because we want to investigate how temperature can affect metabolism or the rate of cellular respiration. So we're going to be using water baths here. And what we'll do is we'll run the experiment at a room temperature, and then we're going to set up a cold water bath here, and we're going to take our organism, whether it's the bean sprouts or the worms, and we're going to put them into this cold environment and run the experiment again. And that way we can see, well, this is the CO2 that was produced at the normal temperature. What's the CO2 rate, the production rate at cold temperatures, and how does that affect the metabolic rate of both the bean sprouts and the worms, the super worms. So I'm going to explain a little bit more on the data tables in another video, but this is the basic equipment that we're going to be using, and I'm going to have to run this experiment basically 4 times. Normally in the lab, I would have 4 groups, and one group would do room worms, one would do cold worms, one would do room sprouts, and one would do cold sprouts, and then we pool our data. But since I'm here in the lab by myself, I'm actually going to set up 2 at a time. So I'm going to have the room temperature running at the same time, and then I'll record the data, and then I'll run the cold temperatures at the same time, and I'll record the data, and then I'll be sharing all of those data tables with you later in the lab.